Hey friends, today we are gonna learn about the dihybrid cross in pea plants. Gregor Mendel observed four types of seeds, and that's when the dihybrid cross experiment came about. They were two yellow seeds, which were either wrinkled or round, or two green seeds, which were either wrinkled or round. So basically, in a dihybrid cross experiment, Gregor Mendel took two parental pea plants, which were of only two contrasting characteristics, which was one had yellow round seed and the other one had green wrinkled seed. So after crossing the parental pea plants, we got two additional characteristics which were not in the original plants. So basically, when it comes to a pea plant, there are millions of characteristics, but Mendel took around only seven of them and experimented on either a pair of them or two pairs, which came about as dihybrid cross. Here you can see seven pairs of chromosomes. Amongst the seven characteristics, the first chromosome had flower color and seed color gene located. But the second and the third pair of chromosomes did not have any of the characteristics Gregor Mendel chose. The fourth pair of chromosome had three characteristics, which were pod shape, stem height, flower position. The fifth chromosome had pod color. The sixth chromosome did not have any Mendel's chosen traits. The seventh chromosome had the seed shaped trait. And in the dihybrid cross experiment, we have taken the traits from the first chromosome and the seventh chromosome, which is seed color and seed shape. So guys, as you can see here, this is the genetic makeup or the pattern of the yellow round plant. So in this plant cell of a pea plant, there are 14 chromosomes in which 7 of them are from the father and 7 of them are from the mother. And two copies of a similar chromosome is known as a homologous chromosome. And hence, like that, there are 7 pairs of chromosomes. During meiosis or gamete formation, all the homologous chromosomes align at the center. As meiosis progresses, the spindle fibers attaches to the center of the chromosome. The spindle fibers then shorten and move towards the poles of the cell. The spindle fibers, as they move, also pull a copy of a chromosome with them to the pole. In the gametes, the number of the chromosome decreases from 14 to 7. In meiosis second, equational division occurs. Hereafter, the cell slowly divides and they form gametes. In a parental cell, the characters are determined by a pair of genes, but in a gamete, the traits or characters are determined by a single gene. And this is the genetic makeup of a green wrinkled seed producing plant. This is the chromosome pattern of a pea plant producing green and wrinkled seed. Just as we saw in the previous parental plant, here also the first chromosome contains gene for seed color and the seventh chromosome contains the gene for seed shape. But the difference is here in the seed color, it will be green and the seed shape will be wrinkled. As we have already seen in the round yellow plant, here too the spindle fibers will attach to the chromosomes and will be moved to the pole. And as they move apart to the poles, they will be divided to 7 pairs of chromosomes. The cell then divides and forms two separate gametes. Here you can see gametes from yellow round and green wrinkled parents. Here for reference, the yellow represents the color and the dark green represents the green color, and the dark orange represents the round shape and the light orange represents the wrinkled shape. Round yellow seed producing plant is crossed with a wrinkled green producing plant, and this cross, all the progenies were round and yellow. This hybrid plant is known as the F1 generation. And when these two gamete fuses, we get a diploid cell. In this diploid cell, we can see characteristics from both the gametes, and there are also 14 chromosomes. Here, the homologous chromosomes can be paired up in 7 pairs. Even though here in the diploid cell we can see both the characteristics, which is yellow and green and round and wrinkled, the phenotype will always be round and yellow. As we know, the first chromosome determines the seed color, and here we have yellow and green, which are the alleles of seed color. Just like that, even in the seventh chromosome, we can see alleles of seed shape, which is both wrinkled and round. From second pairs to the sixth pairs, all the chromosomes are homozygous, which means both the copies have similar characteristics, unlike the alleles. In the next step of this experiment, the F1 generation plants are going to go through self pollination. After self pollination, there will be 4 different types of gametes produced. Now we'll see how the 4 types of gametes are produced. Here you can see 2 types of arrangements. Have you noticed the difference in the arrangement? Here we can see that the green and round is moving to the poles, and yellow and wrinkled are moving to the opposite pole. Here, round and yellow are moving to the upward pole. And here we can see green and wrinkled moving towards the opposite pole. And hence, like this, the parents produce 4 different types of gametes.
Let's look at this scenario. The first gamete is fused with the first gamete of the second parent. Here, both the gametes contain dominant genes. They are round in shape and yellow in color, and the seed will be round and yellow. Let's look at the second scenario. Here, the first gamete is fused with the second gamete of the second parent. Both the gametes contain dominant genes. They are round in shape and yellow in color, and the seed will be round and yellow. Let's look at another scenario. The first gamete is fused with the third gamete of the second parent. Here, both the gametes contain dominant genes. The seed will be round and yellow. Supposing the first gamete is fused with the fourth gamete, here after the fusion, we observe that the cell contains dominant genes for both the traits. So the seed will be round and yellow. Supposing the second gamete is fused with the first gamete of the second parent, here after the fusion we observe that the cell contains dominant genes for both the traits. So the seed will be round yellow. Imagine now the second gamete is fused to the second gamete of the second parent. After the fusion, we find the cell contains recessive genes for seed color and a dominant gene for seed shape. So the seed will be round green. In the next case, the second gamete is fused with the third gamete of the second parent. After the fusion, we see the cell contains dominant genes for both the traits. So the seed will be round yellow. Imagine the second gamete is fused with the fourth gamete of the second parent. Here, after the fusion, we find the cell contains recessive genes for seed color and dominant gene for seed shape. So the seed will be round green. Imagine the third gamete is fused with the first gamete of the second parent. After the fusion, we find the cell contains both the dominant genes for color and shape. The seed will be round yellow. Imagine the third gamete is fused with the second gamete of the second parent. After the fusion, we find the cell contains both the dominant genes for color and shape. The seed will be round yellow. The third gamete is fused with the third gamete of the second parent. After the fusion, we find the cell contains dominant gene for color and recessive gene for wrinkled shape. So the seed will be wrinkled yellow. In this scenario, the third gamete is fused with the fourth gamete of the second parent. After the fusion, we find the cell contains dominant gene for color and recessive gene for wrinkle shape. So the seed will be wrinkled yellow. Fourth gamete is fused with the first gamete of the second parent. After the fusion, we see the cell contains dominant gene for color and shape. So the seed will be round yellow. In this scenario, fourth gamete is fused with the second gamete of the second parent. After the fusion, we find the cell contains recessive genes for color and dominant gene for shape. So the seed will be round green. In this scenario, fourth gamete is fused with the third gamete of the second parent. After the fusion, we can find that the cell contains dominant gene for color and recessive gene for wrinkle shape. So the seed will be wrinkled yellow. Fourth gamete is fused with the fourth gamete of the second parent. Here, after the fusion, we see that the cell contains a recessive gene for both color and shape. So the color of the seed will be green wrinkled. In this chart, you can see the F2 generation of the dihybrid cross. You also can notice 9 plants with round yellow seeds, 3 plants with round green seeds, 3 plants with wrinkled yellow seeds, and 1 plant with wrinkled green seeds. So the phenotypic ratio is 9:3:3:1. One more thing to be noticed here is the factors controlling the traits are inherited independent of one another. Friends, if you like this video, please subscribe it and share it with your friends.